I would like to introduce Professor Cathal Seoighe, who's going to speak in the plenary session. He's the scientific director of the SFI research training in genomics and data science. He started off in physics and then moved to bioinformatics, and he spent time in high-profile institutes in South Africa. And since 2009 has been based in the School of Mathematics, Statistics and Applied Maths in NUI Galway, where he focuses on genomics research. And today he's going to address the theme of genomics revolution and what Ireland can do to join it. Cathal, it's over to you.
Thank you, and thanks for the introduction. So, so for the stragglers, let me share my screen and have a sort of a pretty maybe general overview of the genomics, the possibilities for genomics in Ireland. So for this talk, I thought I would keep it very general because obviously there are people coming from different backgrounds here. So this is just a kind of an overview of what I thought I would kind of talk to you about for those of you who didn't make it into a breakout room or who chose not to be in a breakout room. So on the assumption that there are people here who might not know much about genomics, I'm going to kind of give a couple of minutes of a brief background and refresher, and then talk a little bit about what might be driving revolution in genomics. And I thought I would kind of give a title for this that was kind of fairly general, so open-minded in the sense that, you know, we start by sort of asking ourselves, is there a revolution? And then we'll kind of move on to thinking about, well, what might that revolution brought about by genomics involve? And I'm just going to talk then about some of the kind of healthcare impacts that genomics can have in general, and then think about what genomics could mean for Ireland. And throughout all of this, I've got to also want to declare any potential conflicts of interest of mine. You can see those on my website. I don't have any commercial potential gains from genomics, but like any other academic, might be worth noting that they say that the professors have to have two particular talents. One is to exaggerate the capability to exaggerate the importance of their own field. And the second is to be capable of exaggerating their own importance within that field. So the second won't come into this, but the first you might bear in mind that obviously I come from genomics and am enthusiastic about it. So first of all, the kind of general background and refresher. So we're talking about DNA. This is a kind of a classic picture of the DNA double helix, which people are probably familiar with. And you can boil this down to this kind of code or sequence of letters that represent the different chemicals that are strung together in this molecule that we're seeing here. And this is really the essence of it. And you can think about this, of course, it's a double molecule, but we typically ignore the fact that there's two copies of this running alongside each other because it's enough to just kind of think about this as being at a very basic level, just a kind of like a string or a sequence of letters. And this is what we refer to as these DNA sequences. And of course, there's quite a lot of complexity underlying this whole thing because really these DNA molecules are very long. There's about 3 billion of them in the human genome, and they're all packaged very carefully and tightly around these kind of structures within the nucleus of cells. And how they function can depend on, you know, a lot of factors about how these things are put together and how they work. So there's a lot of details in there that I'm not going to go into, but at a very, very basic level, you can think about a genome as being about 3 billion, or a human genome as about 3 billion of these letters. And then you could ask yourself, well, if there is a revolution in genomics, what might be driving it? And I thought, since we're talking about things that are driving revolutions, it might be interesting to think back to the beginning of the last century and some kind of industrial revolutions that took place, or some revolutions within kind of manufacturing that took place at that time. And if you think, for example, of the Ford Model T car, the Model T and Ford made automobiles accessible to a much, much larger number of individuals, bringing about a kind of revolution for better or for worse in terms of how roads are used and how people get around. But if you think about that Ford Model T from about 1910 to about 1925, they nearly knocked a zero off in terms of the price, the cost of one of these Ford Model Ts, because they just became better at doing things at scale. And you might like to think about, well, what are the kind of equivalent pictures a century later in terms of the human genome? And if you think about the human genome, it cost about 2 billion to sequence the first human genome. And arguably it was kind of finished around 2003. So 2003 with a price tag of around $2 billion, roughly the same in euros. And if you go then 10 years later, almost to where we are today, the price tag has gone down by a huge amount. And you can, so it started off at 2 billion and then it went suddenly to 200 million, or if you knocked 1 zero off it, you'd be at 200 million. That would still be something which would be completely inaccessible to individuals. But if you knocked a further zero off it, and knocking zeros off things is by no means easy, you'd be down to 20 million. And it's if you knocked a further one, you'd be down to 2 million. Another one, you'd be 200,000. Another one, 20,000. You keep knocking zeros off, and we'd have knocked 6 zeros off altogether. And you'd need to then knock off some further price to get to where we are today. So if you going from just kind of a very short period of time, less than 20 years, we've knocked more than 6 zeros off the price of sequencing the human genome. I got mine sequenced about 1.5 years or 2 years ago for about 160 euros or something. So this has become something which is now very accessible and at an individual level. And each time you knock zeros off the cost of something, new things become possible. And I think that's part of what's really driving this kind of revolution in genomics. And at a basic level, you can think about genomes, as I said, as sequences of letters. And these sequences of letters, these sequences of ACs, Gs, and Ts, are responsible for all of the reasons that these two individuals don't look like identical twins. So the main reason that it's become much cheaper to sequence genomes is because, of course, the vast majority of our genomes are all the same. So we can use one sequenced genome to try to help us to work out the sequence of other human genomes. And there's techniques that allow us to do that very quickly. And there's new developments, technological developments, really driving the fact that we can do this now at much greater scale. And when you look at those sequences, as I said, you'll find that most of them are the same. Here in green and red, there are some positions where Michal Martin and Leo Varadkar are different from one another. And what's challenging, of course, with these is the fact that there's only going to be a relatively, well, there's only roughly one letter out of every 1,000 or so where these people will differ, but that still amounts to several million of these letters where they differ. And what's challenging is that most of these differences don't actually make any difference. So they don't contribute, or probably don't contribute, to the differences between these guys in terms of their susceptibility to diseases, etc. So what's really challenging is to find these places where there are differences that do actually make a difference. And if you think about how genomes differ in general, well, there are inherited differences. So most of these differences between Michal and Leo are going to be differences that they've inherited from their parents. There's going to be new mutations, which these guys are, which are unique to these guys. So they're mutations that occurred in the kind of the germline, or the germ cells leading to these individuals. And only quite recently have we been able to actually directly measure how many of these new mutations an individual has. And so if you take nothing away from this meeting, and if you end up with, if you end up wanting to make sure you have no regrets for staying in the main room rather than going into the breakouts, well, maybe one thing you might have learned from today is that each one of us has probably less than about 100 of those. So probably typically around 60 or 70. That's a kind of an interesting figure to lodge in the back of your mind. So you, unique to you, probably have about 60, 70, 80 mutations that occurred in the line leading to you that are unique to you. And you can think of these almost like kind of lightning strikes in the genome. Most of them won't do anything to you, but sometimes they do. And understanding how they might affect people can be really important. And then lastly, there are mutations in ordinary cells. These are by far the most common, and these are in the field of genomics called somatic mutations. You have trillions of cells, and each of those cells can have mutations. And these mutations are not just a curiosity, they're really important because these are what can lead to cancer. So cancer is a disease of the genome, and most cancers, many cancers at least, arise through mutations that occur in your ordinary cells and enable those ordinary cells to escape from the controls that the body normally places on them and effectively to go rogue and develop a tumor. So all of these different types of mutations are really important, but potentially for different reasons. And then what is genomics? Well, genomics is not just about sequencing genomes. Genomics is really all of the kind of genome scale approaches to biology. So because it became possible to sequence a genome, people started to think about things in a very different way. So we now think about things in a kind of a global way. So genomics really heralded a kind of a change in perspective about the way we approach biology. We now want to approach biology in the way we want to understand the sequence of the entire genome, the sequence of all of the genes, the function of all of those genes, and we measure them in lots of different ways. And lots of the different ways in which we measure what genomes are doing work or are based on these basic sequencing technologies that have become really cheap and effective. So genomics is a whole field, a whole slew of related technologies that are really taking a very ambitious approach to how we understand biology at a very detailed level. And then a genomics revolution has been, the genomics revolution has essentially been an era of extremely rapid progress in biomedical research, and more recently, the adoption of similar approaches and the insights derived from this rapid progress in biomedical research into medicine. So the adoption of new approaches in medicine based on genome sequencing and other genomics technologies. And this has enabled medicine to become much more individualized, or at least has really advanced the prospect of implementing kind of personalized medicine, or medicine at the individual level, that takes account of the differences between individuals and tries to sort of treat and understand disease risk at that individual level. And what I'm going to do now, as I said in the outline, is just cover a few of the different areas in which this new approach to medicine has had an impact. And these are in no particular order, but I thought I'd start first with understanding the prediction of risk. So for many years, we have had good knowledge of specific mutations that confer risks of certain diseases. And there's very well-known examples of that. For example, cystic fibrosis. If you have the mutation, if you have 2 copies of that mutation, you may have the disease. And these are individual mutations with a big effect. And these are, of course, really important for the individuals who are affected by them. But many diseases, or possibly even most common diseases, have a strong genetic component, but they're not attributable to one gene, and they're not attributable to one mutation. So these are called polygenic diseases because there's many, many mutations that contribute to them. And the genomics revolution, the genomics era, has made it possible to understand how these mutations collectively confer risk. So people are able to sequence genomes, genotype an individual, and understand where that individual lies in this kind of risk profile for some of those polygenic diseases. So these are really important diseases. They could be things like type 2 diabetes and other metabolic disorders, and even the risk of certain types of cancer, certainly heart disease, and lots of other kind of diseases that have a very strong genetic component, where that genetic component is distributed over lots and lots of genes in the genome. So this is really important because several of those diseases are modifiable. So if you can identify the individuals who are at particularly high risk, you can engage with those individuals and help them to reduce their risk. So this is an area of ongoing developments, not without its challenges, particularly in terms of understanding how this risk prediction works in different population groups. So an area of still rapid development. There's rare disease diagnosis. You remember those lightning strikes in your genome. Some individuals are born, unfortunately, with lightning strikes that have affected, or mutations that have affected really important genes. And these individuals are sometimes born with a condition which looks like it may be a sort of a genetic condition, but there's no family history. And one of the things that can be done in this case is, if you sequence the genome of both parents and of the affected infant, you can often identify where were those new mutations that occurred in this infant, and which are the ones that are most likely to have had an impact. And this can be a huge boost in terms of helping people to understand what the origin of that disease is, in some cases helping to identify treatments. This is being done systematically in some places. In Ireland, it's not being done systematically. It's being done currently pro bono by Genuity Science, but I'm not aware that this has been made systematic in the healthcare system, but certainly should be something which is part of what the state should provide, in my opinion. And Prognosis is another area that genomics in general has had huge impacts on. And here in this case, maybe I might illustrate this by example. So I was discussing this with the head of the Bioinformatics in St. Jude Children's Hospital in the US, in Memphis in Tennessee a couple of years ago. And already a couple of years ago, they were routinely sequencing the genomes of all of the infants, all of the childhood cancer patients that were appearing for treatment in that hospital. So this is a really high-end hospital where they have very advanced technologies, but they had already appreciated there, even a couple of years ago, that it's extremely important to be able to apply genomics for these patients, mainly because it can be extremely useful for prognosis. So when children are treated for childhood cancer, those treatments can in some cases have long-lasting side effects. And so it's really important to calibrate the treatment appropriately to the severity of the prognosis of the child. So if a child has a good prognosis, you want to calibrate, you want to treat that child less aggressively than a child who has a poor prognosis. And this is extremely important because it can reduce the impact, in some cases, life-lasting impacts of the treatments for some of the children. So in already a couple of years ago, this was found to result in what's referred to as actionable insights for the majority of patients. So for the majority of patients, getting all of these genomics assays was providing some concrete information about how these children should be treated. And so that's extremely important. Not building that into our healthcare system would be a dereliction of duty, the duty of care that we have to provide the absolute best possible treatment for patients with childhood cancers, and to calibrate those treatments according to the disease that they have and the severity of the disease. So this is something that I think is really important as we begin to reform or build a new children's hospital. We should begin to think about how incorporating these techniques to maximize to optimize the treatment of these patients. In general, for disease treatment, there are many cases where drugs have been found to have genetic interactions. So this is referred to as pharmacogenomics, where there are drug treatments that depend on the condition, that depend on the genome. So some people should take a smaller dose of a particular drug, some people should take a larger dose. In particular, again, in cancer, there are several treatments that now have what are called companion diagnostics based on genomics. So these are cases where the treatment that you give depends on the outcome of a genomics assay. And these are things like some of the new immunotherapies in cancer. And again, the best way to treat these people is to, again, ensure that the treatment is appropriate to the genetic background of the individual. And again, to give the best treatment we possibly can to Irish patients, we should make sure we're doing that. There are new ways of detecting cancer early by blood biomarkers. So one of the really interesting areas of current development is to look at what tumors deposit into the bloodstream. So tumors deposit proteins and also DNA fragments into the bloodstream. If you take the blood and sequence the DNA fragments, you can sometimes identify the mutations. And remember, we talked about mutations in ordinary cells giving rise to cancer. If you can sequence the fragments of DNA in the blood that have been shed from the tumor, you can sometimes identify that this individual has a cancer, and also go further than that and even identify the site of the cancer. Again, that's extremely important because cancer treatment is all about catching cancers early. If you can catch cancer early, it's much more treatable than late cancers. And then just obviously the current era that we live in has been an era that's been dominated by a viral infection, which has become pandemic. And genomics doesn't apply, of course, only to human genomes. We're not the only things that have genomes. Every living thing, and even things that are debatable whether they're living or not, like viruses, have genomes. And sequencing the genomes of pandemic coronavirus has been extremely important in terms of tracking the spread of the disease and also tracking the spread of variants that are really the current major threat to the reopening of the global economies. Okay, and so with all of these possibilities, all of these things that we can do with genomics, how can Ireland actually benefit from that? And I realize I'm kind of running a bit short in time, so I'm going to go through this quite quickly. But
you have a few minutes.
Okay, I have a few minutes left, so I'll talk a little bit. I want to leave a little bit of time in case anybody has questions as well at the end. So firstly, there's an urgent need for a government coordinated national strategy. And we all heard from Arno Pilotti how the government and multiple different departments came together to ensure that the Finnish people could gain the access to genomics and the benefits, the potential benefits of genomics. Ireland hasn't really done that yet, and there is an opportunity to do that. There's certainly a need to do that. So there's a need for the government to become actively involved, and not just one group within one department, but ideally a government-wide initiative. And it's interesting to see not just Finland, but other countries where this has been very successful. There has been strong leadership. The UK is also another good model. There's been very strong leadership at the top level from the UK. And again, very, very strong coordination from the government. So that's urgently needed. But of course, it's not just up to the government. That movement should involve everybody who has a stake in this: clinicians, scientists, public health doctors, patient groups. Events like today are really important. Getting everybody who has a stake in this to participate, to contribute also to the kind of the pressure to ensure that this is developed. Health economists have an important role. People who understand health systems have an important role in how this should work. And as we heard also from Arno, there's a need for a great deal of thinking around how the rights of individuals are protected within these kinds of projects. And that includes protection of data, clear legislation to protect the rights of patients across the full spectrum of patient rights. But it's, of course, not sufficient to only protect rights. We also want to reap the benefits. And we want to have that right balance, to achieve that right balance between, on the one hand, of course, protecting all patient rights, but also ensuring that we can advance the implementation of genomics towards the access of precision disease management for the people of Ireland, because we want to give the best treatment and the best management of disease possible in Ireland, and we want to ensure that the contribution that genomics can make to that is fully realized. And part of that process is going to involve stimulating and managing appropriate public-private partnerships. And again, from the FinGen example, we saw all of the different partners involved. There are hospitals involved, but there are also several companies that have been funding what they've been doing in Finland. And this is the typical scenario. We have, for example, again, I mentioned the UK, very strong public-private partnerships in the UK, but very often government coordinated and government managed. And so the stimulation and management of those partnerships is a really important role, largely of government in this process. And this needs, in the Irish context, to be connected with the broader reform of the health system, which is currently underway. So Ireland is in a process of reform of its health system, which many would feel is badly needed. That's a good thing in the sense that reform provides opportunities to do things better. And we should, of course, not at this stage be trying to replicate what's been done elsewhere, because a lot of advances have already been made. If we look at other examples around the world, we may be able to leapfrog some of that progress and build on some of that progress and think about how that progress, where that progress is going to lead to, and how that could be integrated and incorporated with our healthcare system as it gets reformed. And lastly, if you want to have a look at some maybe a more detailed description or discussion of some of these issues, Walter Koch at UCD and I and a whole bunch of others have written an article in HRB Open Research discussing a strategy for genomics in Ireland and how that might contribute to the development of improved healthcare in Ireland. So you can have a look at that article, which is open access there. And if there are any questions, I'd be happy to take them. Thank you.
Thanks so much, Cathal. We do have a few minutes for questions, and I even have some comments in the chat room from people saying they're glad they stayed for this session. So well done. And I know you talked through a lot of themes around the importance of genomics for rare diseases, and the comment made about there's some pro bono work being done on children who are diagnosed with rare diseases and sequencing of their genome. And it's not part of the systems approach, to genome sequencing at the moment, whether it's children with rare diseases or children with cancer, or people with tumors who want to get their tumor sequenced. Very often they're paying for it themselves. So, from your perspective, how important is it for the infrastructure within our health system to enable that service for patients?
I think it's really important. I don't think we should be relying on the kind of pro bono contributions from companies. I think it's great that they're doing that and that they've done that. I think there are families who have benefited from that. But I think that's okay up to a certain point, but you really need to take it to the next level and make sure that it's provided in a way that's systematic, so that to ensure that if this is the kind of approach that is the optimum approach for people with these kind of diseases with kind of suspected de novo origin, that is made available to all of the children who would benefit from it, and that's guaranteed and underwritten by the state, I think is really important.
And it just like in terms of the staff, the nurses, the doctors, the technicians, the pharmacists, do we have a workforce who are trained in genomics? I know the answer to this, but I'd love to get your perspective on that. Like, are our people in our health system ready for this as well?
No, I think we need a lot more, and that's something that's a particular interest of mine, actually, as the scientific director of a Centre for Research Training in Genomics Data Science. I mean, I think that's really one of our objectives, is to try to think about, well, what is the kind of future workforce that's going to drive this, and can we make a contribution into ensuring that that future workforce is available? Do we have all of the people who would be required? Do we have those today? No. But that's an opportunity. I think it's an opportunity to think about skills development. It's also a reason why this is a kind of there's a need for a whole of government approach, because we shouldn't just think about this as something which belongs to the Department of Health. We should think about it as being the Department of Higher Education should be in skills development. Higher education comes into this, and the Department of Enterprise also comes into it. It's really a whole of government approach that would be required, partly because it's complex to ensure that we can provide this kind of service and these kinds of treatments.
Absolutely. And I know there's a shared island document in the Department of the Taoiseach, and the second strand of that is about research. So I do wonder if this should form part of that on an all-island basis.
And absolutely, you're absolutely right. It should. Yeah. And there's great opportunities there. There's fantastic work being done in the north of Ireland as well, and of course links in very nicely with what's happening in the UK.
There's a very interesting question from one of the listeners about the future proofing by having multi-omics approach like epigenomics and others around the single cell genomics. Is the focus on genomics only in the first instance with a view to expand genomics national strategy at a later date? So are we starting, in one place and hoping to reach out beyond that single strand?
So in genomics, and I think this becomes down to kind of questions of definition sometimes. So in genomics, I would include epigenomics and single cell genomics. So I would kind of use that term quite broadly. And I kind of tried to make that point in the talk there, where you kind of, all of these kind of techniques that are enabled by genome sequence technologies and that kind of genome scale kind of perspective, I would include within genomics. So I think we should be very open-minded about those kinds of terms and how those terms are used.
What do you think about, what I talked about earlier, we do have at the moment, science is ahead of where our system is, and what's available to patients in the clinic today. So very often patients are going out there and they're getting their own, and I see that in cancer, getting their own sequencing done. And then they're rocking into their oncologist with a big file and saying, "This now, can you just find the drug for that, please?" And that happens a lot. And I suppose we have a real responsibility, don't we, in terms of communicating how it works, how that precision medicine piece works. And, within Ireland, some of the BRAF mutations, we're doing work within trials, but it's not kind of part of, the systems approach for patients. So you can understand why patients are going out there and doing it for themselves, but there's a very difficult communication then with the patient and their families when there isn't a treatment for the target that's found.
Yeah, there's certainly challenges there, and but I think what we need to ensure is that I think the system will always be a little bit behind where the science is, and we need to be sort of like, I guess, sort of accept that there is going to be a certain lag. But I think it's extremely important that our systems and our health systems are not lagging behind where the best health systems in the world are. So we should look at, well, what are the kinds of treatments that are being routinely provided in other countries, and are we at least at that level? And then we continue to be pushed by the science, and I think that's a good thing, that the science continues to kind of challenge the way things are currently done, because it creates possibilities and creates ways of doing things in the future that are better.
Yeah. I suppose from your perspective in bioinformatics, does it frustrate you that we don't have an electronic health registry? Just listening to Arno's presentation and the linkages between, you know, the 500 plus with their health information, you kind of be envious looking at that.
Yeah, absolutely, it does. That it does frustrate me. I think it is a big disadvantage not to have that. I think it's something we should be working on, and it's something that's, you know, it should be very possible to address, and it would enable things to be done much better, I think.
Yeah, like it is important, isn't it, to be able to connect the genomic data with the life story of a patient's health journey.
It's extremely important, and I think that more and more people are realizing over the last few years that really good information about health outcomes, about phenotype, about drug treatments, this is extremely important, because the sequencing itself has become sort of less and less expensive and less part of things. Where the gulf is in terms of the analysis, in terms of being able to connect the genome with the different health outcomes, different treatments, and different responses to treatment.
Great, Cathal, we kept talking there because we were awaiting the return of the foot soldiers in the breakout groups. So I want to really thank you. I've been through a lot of these presentations, but you really made it simple especially for those like me who are not scientists and didn't start off that way. Thank you very much. And I think there's your last slide really needs to go into this report, because it's very key in terms of the recommendations, you know, from the national plan to the importance of genome sequencing and access to it for our population. So thank you.